Human history has long been written in stone and bone, a story told through the fragments of what remains. But a new era has arrived, one where the very molecules of life are resurrecting the voices of those long forgotten. We are beginning to see the past not as a series of static artifacts, but as a living, breathing code. This is the story of how we are doubling our understanding of human history in just a few short months. From the massive industrialization of ancient DNA to the personal secrets of a family buried 12,000 years ago. We are mapping the real-time evolution of our species across 10,000 years of survival, even reaching into deep time to reevaluate the very first traces of animal life on our planet. Welcome to the molecular resurrection. The story of our world is about to be rewritten. In June 2026, the field of paleogenomics reached a historic tipping point that fundamentally changed the scale of research. The amount of available ancient DNA data globally has effectively doubled in a single massive effort. Led by the Reich Lab at Harvard, this industrialization of DNA has mapped over 16,000 individuals, providing us with the first high-resolution map of human life spanning 10 millennia across Europe and the Middle East. This scale of data allows scientists to move beyond broad migration patterns and see something more profound. We are now watching evolution happen in real time, within the human body, in response to a changing world. The success of this project relied on a new computational method developed by researcher Ali Akbari. This math allows scientists to distinguish the true signal of natural selection from the noise of migration and genetic drift. For the first time, we can pinpoint exactly when and where specific genes began to change in our ancestors. We've identified nearly 500 genetic loci under strong directional selection over the last 10,000 years. These are the evolutionary scars, the adaptations that allowed our ancestors to survive in new environments. As we shifted from hunting and gathering to farming, our bodies were forced to change in response to new diets. The rise of celiac disease is a lingering legacy of the Neolithic Revolution, a genetic risk we still carry today. We also see shifts in body fat storage, likely an adaptation to the changing agricultural diets of our ancestors. Even cognitive and psychological traits show signs of selection, linked to performance and modern health challenges. As our ancestors began living in dense settlements, our immune systems faced an onslaught of new pathogens. The variants we now link to autoimmune conditions may have once protected us from the plagues of the early Neolithic. We are watching human biology change in response to culture and environment, step by agonizing step. In southern Italy, the 12,000-year-old Grotta del Romito burial has become a genetic cold case that speaks to the soul. Originally discovered in 1963, these two individuals were found interred in a poignant, lasting embrace. This suggests that oxygen levels in ancient oceans were lower than we thought, delaying the timeline for when the first mobile animals appeared. Genetic analysis has confirmed a mother-daughter bond and the world's earliest genetic diagnosis. Romito too suffered from acromosomtic dysplasia, a rare condition causing severe limb shortening. Standing only 3 feet 7 inches tall, she was far shorter than her mother and the average of her peers. Extraction of DNA from the petrous part of the temporal bone was the key to unlocking this ancient family secret. This dense bone acts as a vault, protecting fragile DNA from the ravages of 12,000 years. Her survival into her teens in a harsh Ice Age environment proves that her community provided sustained care. Despite severe mobility limitations, she was supported and valued, a testament to prehistoric compassion. This discovery proves that empathy isn't a modern invention. It is a fundamental part of our shared human code. Even 12,000 years ago, we took care of our own, preserving the dignity of every member of our society. Then there is Lola, who left no bones but left her entire biological profile in a piece of prehistoric chewing gum. Reconstructed from birch pitch, she was a hunter-gatherer from Denmark who was lactose intolerant and suffered from ancient gum disease. Trapped in the pitch was the record of her last meal, mallard ducks and hazelnuts, and her entire oral microbiome. Lola carried the Epstein-Barr virus and gum disease, giving us a personal health profile from the dawn of civilization. Like most Europeans of her era, she lacked the variant for lactase persistence, confirming she could not digest milk. Through a single sticky artifact, we can reconstruct the phenotype and health of a woman from 5 millennia ago. We can now reconstruct a whole life and a whole world through a single sticky artifact trapped in the soil. Lola's story has established birch pitch as a critical bioarchive, protecting molecules that would otherwise degrade. Beyond human history, we are reaching into deep time to reevaluate the very first traces of animal life on Earth. Studies of microfossils in Brazil, once thought to be animal tracks, are being reclassified as ancient bacteria and algae. This suggests that oxygen levels in ancient oceans were lower than we thought, delaying the appearance of mobile animals. Every discovery we make with these molecular tools is a new chapter in the epic story of life's resilience. In the sands of Egypt, a newly discovered jawbone is shifting the cradle of primate evolution to North Africa. Fossil remains analyzed using NCBI's tools suggest that modern apes may have evolved further north than previously theorized. This rewrite of our evolutionary timeline shows that the story of our origins is more epic and far-reaching than we knew. In a crossover between physics and biology, researchers have reported the first observation of the Migdal effect. Predicted in 1939, this discovery has deep implications for how we understand light-dark matter and its interaction with life itself. The recoiling nucleus ejections provide a new way to sense the invisible forces that surround us in every moment. We are learning that the fundamental laws of the universe are etched into the very biology of our existence. Biophysicists are now using the Migdal effect as a high-precision tool for single-molecule intracellular imaging. By tracking ion transport at sub-nanosecond scales, we can observe the missing mechanics of cellular signaling. This technology has led to the development of quantum biotrackers that can monitor the health of our internal barriers. These trackers provide an early warning system, detecting breakdowns in our lining long before physical symptoms appear. NCBI has also added the nitroplast feature to its database, marking a major milestone in the evolution of plant life. This nitrogen-fixing organelle represents a fundamental bridge between bacteria and eukaryotes, a discovery that changes how we view the evolution of plant life and our planet's nutrient cycles. The nitroplast is only the fourth recorded instance of primary endosymbiosis in Earth's entire history. Evolving just 100 million years ago, it is a much more recent event than the origin of mitochondria. Scientists have identified over 350 proteins specifically tagged and imported into the organelle. This genetic handover is the definitive hallmark of an organelle, proving it is a permanent part of the cell's machinery. The goal now is to integrate nitroplast-like functions into terrestrial crops, bypassing the need for synthetic fertilizers. By engineering plants that fix their own nitrogen from the air, we could drastically reduce global agricultural emissions. In the modern era, molecular biology is transforming how we fight disease through precision oncology. Identifying markers like MHC2 allows us to predict patient responses to life-saving immunotherapy with high accuracy. Tumor cell intrinsic expression shapes local niches, making some cancers more susceptible to modern treatments. The Lancet published breakthroughs in lung cancer, showing significant improvements in overall survival rates. This combination is outperforming previous standards of care, marking a major step forward in treatment. New targeted therapies like daraxonrasib are showing superior outcomes for metastatic pancreatic cancer. This precision-first approach is offering hope where standard chemotherapy has previously failed. In neurology, we are unlocking the signatures that protect the human brain from cognitive decline and Alzheimer's. Specific protein signatures in these remarkable individuals may hold the key to lifelong brain health. And we are finding non-invasive ways to diagnose conditions like Parkinson's years before the first symptoms appear. Tools like earwax evaluations could change the course of treatment by allowing for much earlier intervention. Research in brain metastasis revealed that radiation can actually enhance T-cell-mediated immune responses. By priming the brain microenvironment, radiation may make checkpoint inhibitor therapy far more effective. PubMed has also integrated millions of new citations, refining how we index and retrieve the world's medical knowledge. Ensuring that the world's knowledge is accessible to everyone, from the clinician to the curious researcher. Our global health depends on real-time mutation tracking. NCBI virus now monitors outbreaks with speed and accuracy. This real-time data ensures that the world can respond quickly and effectively to emerging infectious threats. Developments in mucosal vaccination could revolutionize how we prepare for future respiratory pandemics. Offering broader protection than traditional injectables, these vaccines are a major step in global preparedness. The NIH is investing heavily in human-based research models, moving away from traditional animal testing. This shift underscores a move toward more accurate and ethical science, tailored to human biology. The launch of the Center for Phage Therapy underscores the shift toward AI-driven antibiotic alternatives. By using AI to identify effective phage combinations, we are finding new ways to fight drug-resistant infections. In rheumatology, targeted treatments like nipocalimab are showing promise for severe Sjogren's disease. This paves the way for precision treatments based on individual autoantibody and IgG levels. Moving complex cancer treatments to an outpatient setting is saving thousands of hospital days globally. This 85% reduction in length of stay is a victory for patient quality of life and healthcare systems. As GenBank grows to 50 trillion base pairs, we are doing more than archiving data. We are building a library of life, ensuring that every discovery is linked to its geographic and historical context. preserving our history for the future. New bioarchaeology standards ensure that every genetic sequence is permanently linked to its archaeological origin. The molecular resurrection is as much about accurate preservation as it is about groundbreaking discovery. Nearly 54 trillion bases of life's code are now stored in GenBank, a treasure trove of biological secrets. This data reveals the biological secrets of every corner of our world, from the depths of the ocean to the peaks of the mountains. PubMed has also integrated millions of new citations, refining how we index and retrieve the world's medical knowledge, ensuring that the world's knowledge is accessible to everyone, from the clinician to the curious researcher. NCBI is also evolving its infrastructure, retiring old protocols for more secure and efficient data delivery systems. These updates ensure that researchers can access massive repositories of genomic data with speed and security. Every gene we map and every story we recover is a testament to our shared human journey and our resilience across time. We are no longer just studying the past. We are bringing its voices into the present, solving mysteries and saving lives. The molecular resurrection has only just begun. The code of life is waiting to reveal its next epic chapter. Modern medicine is pivoting toward mucosal immunity, stimulating protection directly at the site of entry for viruses. By targeting specialized M cells, new vaccine platforms can transport antigens directly to the mucosal immune system. These vaccines stimulate secretory IgA, which provides a first line of defense that traditional shots cannot match. This approach has shown a 45% increase in sterilizing immunity, preventing transmission entirely. To bridge the translational gap, researchers are now using human lung and intestinal organoids for testing. These models allow us to see how the first exposure to a pathogen determines our long-term immune response. By understanding the pathways of entry, we can tailor our defenses to the specific biological terrain of the human body. We are watching the birth of a new era in vaccinology, where protection is as precise as the code it defends. Inspired by the nitroplast, synthetic biologists are now attempting to create custom organelles for human cell lines. These organelles could perform metabolic functions like producing therapeutic proteins directly within our bodies. The discovery of the nitroplast is a blueprint for how complex life can be re-engineered and enhanced. The boundaries between fundamental science and applied medicine continue to blur, revealing a future of endless possibility. Every gene we map and every story we recover is a testament to our shared human journey and our resilience across time. From the sands of Egypt to the laboratories of today, our story is more connected than we ever knew. As we continue to unlock the secrets of our past, we are building a healthier and more compassionate future. Special thanks to the Reich Lab, UC Santa Cruz, and the researchers at the Grota del Romito. Our journey through the molecular history of life is just beginning. What will the next chapter reveal? Stay curious, stay human. The code of life is all around us. Thank you for watching. A New History of Life. The Molecular Resurrection. End.